Transduction is a gene transfer method where viruses called bacteriophages transfer genetic material from one bacterium to another. They use bacterial DNA to infect a new host cell by injecting it into the genome. An example of this is E. coli.
Step 1. Infection. The bacteriophage infects the bacteria cell by attaching itself to a bacterium and infecting the host cell. Step 2. When a phage takes over a bacterium, it essentially hijacks the bacterium's cellular machinery, breaking the bacterium's DNA into fragments. The viral DNA instructs the bacterium to produce new phage components, DNA and proteins, assembling multiple copies of the bacteriophage inside the bacterial cell.
Step 4. During the assembly of new phages, sometimes fragments of the bacterial DNA instead of the viral DNA are mistakenly packaged into the phage's protein coat. Step 5. The bacterial cell is lysed, releasing the newly formed phages into the surrounding environment. Some of these phages will contain bacterial DNA instead of viral DNA. Step 6. The new phage carrying bacterial DNA can then infect another bacterium. However, instead of injecting viral DNA, it injects the bacterial DNA fragment from the previous host.
Step 7. The bacterial DNA injected into the new cell combines with the recipient's genome. This results in the transfer of genes from the first bacterium to the new one. Step 8. The transferred genes are then expressed in the recipient bacterium, giving it new traits such as antibiotic resistance. Aftermath of transduction. When these genetic fragments are transferred to a new E. coli host, they can introduce specific traits such as antibiotic resistance genes, increased virulence factors, or new metabolic function. This process speeds up E. coli evolution, helping the bacteria quickly adapt to antibiotic.
A real-world example of this is that E. coli is susceptible to most modern medicine, yet is able to adapt and become resistant to these antibiotics. Since E. coli acts as a donor and recipient of resistance genes and adapts to new antibiotics by obtaining or transferring genes to neighboring cells.